Welcome to our deep dive, into the fascinating world of Clostridium species. We're going to be using Principles and Practice of Clinical Bacteriology, 2nd edition, by Gillespie and Hockey as our guide. As medical microbiologists, you're probably already pretty familiar with these bacteria and the range of diseases they cause. But we'll be digging into some of the key information that you need to know.
And I think what's really interesting about Clostridia is that they're practically everywhere, but they can cause some really serious problems. I mean, they're amazing at surviving. They can just hang out as these hardy spores until the conditions are right, and then boom, they emerge.
Yeah, like those time capsules, waiting for the right moment to spring back to life. So let's start with the basics. What exactly makes a bacterium a Clostridium?
Okay, so these are gram-positive anaerobic spore-forming rods. And that combination is really important. It's what lets them survive in all sorts of environments you wouldn't expect.
Which explains why we find them everywhere.
Right.
Soy water, even in our own guts.
Exactly. And because they're so diverse, they play a lot of different roles. A lot of them are harmless saprophytes breaking down organic matter.
The recyclers of the bacterial world.
Yeah. And some are even commensals just living peacefully inside us.
Our little microbial roommates. But then, of course, we have the pathogenic ones, the troublemakers.
Right. And that's where things can get a little tricky. Identifying Clostridia can be tough. Some can tolerate oxygen better than others. And their metabolic byproducts, well, let's just say some of them can be pretty smelly. And then there's the whole classification issue, trying to balance scientific accuracy with what's practical in the clinical setting.
It's always a balancing act, isn't it? So we've got this diverse group of bacteria, some helpful, some not so much. Let's focus on the ones that are most important to us in healthcare. And I think we have to start with the one that's on every clinician's mind, Clostridium perfringens, the notorious cause of gas gangrene.
Oh yeah, C. perfringens. It can turn a wound into a life or death situation so quickly. Rapid diagnosis and treatment are absolutely critical.
Absolutely.
Yeah.
So let's talk about what makes this one so dangerous. What are its key characteristics?
C. perfringens is a large, rod-shaped bacterium. And it's interesting because it doesn't form spores easily in the lab. But in the right conditions, like damaged tissue, it can really take off.
And that's when it starts releasing those toxins, right?
Yeah.
It's always amazed me how such tiny organisms can produce such powerful substances.
I know, right? C. perfringens actually produces a bunch of different toxins, but the most important one is alpha toxin. It's a phospholipase C, and it mainly targets phosphatidylcholine, a major component of cell membrane. So basically, it attacks the foundation of our cells.
Like a demolition crew just tearing things down. No wonder it causes such rapid tissue destruction.
Exactly, and that rapid tissue destruction is what makes gas gangrene so dangerous. It creates this anaerobic environment, which is perfect for the bacteria to multiply and release even more toxins. And then as the tissues break down, you get that characteristic gas production.
A vicious cycle. So you've got the contaminated wound, the bacteria growing, toxins being released, gas building up. It all happens so fast.
It does. Gas gangrene is known for its rapid progression. And if it's not treated quickly, it can lead to systemic toxicity and even death.
Which is why recognizing those clinical features early on is so important.
Absolutely. The classic signs are pain that seems out of proportion to how the wound looks. The area gets swollen and tense. You can often feel crepitus, that crackling sensation from the gas. And the tissues might look pale at first, but then they turn this dusky bronze color.
Definitely a clinical picture you don't want to miss. And speaking of toxins, C. perfringens can also cause food poisoning, right?
It can, but the mechanism is different in that case. With gas gangrene, it's those toxins being released directly into the wound. But with food poisoning, it's mostly about a specific enterotoxin, usually produced by type A strains.
So you eat some contaminated food, and then what happens?
The bacteria sporulate in your small intestine, and during that process, they release this enterotoxin.
And unlike alpha toxin, this enterotoxin doesn't destroy tissues directly, right?
Right, it targets the cells lining your intestines. Basically, it causes those cells to die, disrupts the gut barrier, and triggers inflammation. And all that leads to diarrhea, sometimes really severe diarrhea.
Not a fun experience. Okay, so we've covered the dangers of C. perfringens. What about C. novii? I know it's been making a comeback, especially in certain groups.
Yeah, C. novi. It's another large, rod-shaped bacterium with oval spores, and it's a strict anaerobe, even more so than C. perfringens. We've been seeing more infections lately in people who inject drugs.
I've heard about outbreaks linked to contaminated heroin. What makes this group more vulnerable?
When you inject drugs, especially intramuscularly or subcutaneously, you're basically creating this low oxygen environment that C. novi loves. And the injection itself can damage tissues, which just makes things worse. It gives those spores a chance to germinate, and the bacteria can really take hold.
And like C. perfringens, C. novii has its own set of toxins, right?
It does. The alpha toxin is the main one, and it's pretty potent. It's actually similar to some of the toxins produced by other Clostridia, like C. difficile and C. sordelii.
So there are some common themes running through these pathogenic Clostridia. What does this alpha toxin do? What does it target?
It basically attacks the cell's cytoskeleton, causes those internal structures to collapse, so the cells round up, stop working properly, and eventually die. You get severe tissue damage, and if the toxin gets into the bloodstream, it can be fatal.
Sounds like early and aggressive treatment is key. But before we get to treatment, let's talk about another Clostridium species that's infamous for its toxin, C. botulinum, the producer of the most potent neurotoxin we know of.
Oh, C. Botulinum is a fascinating one. It's not actually a single species, though. It's more like a group of different types. But they all have one thing in common. They produce botulinum neurotoxin. We call it BONT.
Like a family of assassins, each with its own specialized weapon.
Perfect analogy. And these neurotoxins are incredibly potent, even in tiny amounts. They target the nervous system, specifically the neuromuscular junction.
The point where nerves and muscles communicate. And that's where the paralysis comes in, right? That classic descending paralysis we see in botulism cases.
Exactly. BONT blocks the release of acetylcholine. That's the neurotransmitter that carries signals from nerves to muscles. No acetylcholine, no muscle contraction. And you get that flaccid paralysis.
And what's interesting is that C. botulinum has different ways of delivering this toxin, which is why we see different forms of botulism.
Right. The main categories are foodborne wound and infant botulism. The route of transmission is different for each, but the end result is the same. That disruption of nerve-muscle communication and paralysis.
So let's start with the classic presentation, foodborne botulism. What are the common culprits there?
Usually it's improperly home-canned foods, especially low-acid vegetables and meats. The bacteria can grow in those sealed jars, produce the toxin, and then someone eats it without knowing.
And even a tiny amount of that preformed toxin can cause serious problems.
Oh, yeah. Wound botulism is less common, but we are seeing an increase in cases, particularly among people who inject drugs.
The contaminated needles and the lack of oxygen at the injection site create a perfect environment for C. botulinum, I guess.
Exactly. It really highlights the need for harm reduction programs. And then there's infant botulism. That happens when infants ingest C. botulinum spores. Their gut microbiome isn't developed enough to fight them off, so the bacteria can colonize and produce toxin.
Which is why honey is off limits for babies under a year old.
Right. Honey can contain those spores. So 3 different routes of infection, but all leading to the same serious outcome: paralysis caused by botulinum neurotoxin. We should probably talk about tetanus, too. It's another neurotoxic clostridial infection, but it presents very differently from botulism.
Oh, yeah, tetanus.
Yeah.
Caused by C. tetanus.
Yep.
It's all about spastic paralysis, right? Not
Yeah.
flaccid paralysis like we see with botulism.
Exactly. Tetanus is a really serious disease, often with really painful muscle spasms, and it all starts with those C. tetani spores getting into a wound.
And those spores are tough. They can survive in the environment for years just waiting for the right opportunity.
Yep. Deep puncture wounds that are contaminated with soil or debris are especially risky.
So the spores get in, they germinate in that nice anaerobic environment, and then they start producing tetanus neurotoxin. We call it TENT. But unlike botulinum toxin, which acts at the neuromuscular junction, TENT travels.
It travels through the nervous system all the way to the spinal cord, and there it blocks the release of inhibitory neurotransmitters,
Which are?
glycine and GABA. They're essential for controlling muscle activity. They're like the brakes that prevent overstimulation and uncontrolled contractions.
So TENT basically cuts the brakes on muscle activity.
Exactly. And without those inhibitory signals, the muscles go into overdrive. That's what causes those classic tetanus symptoms. Muscle rigidity, those painful spasms and trismus where the jaw muscles lock up.
A terrifying but fascinating disease. And like wound botulism, we're seeing a rise in tetanus cases among people who inject drugs.
It's the same risk factors: contaminated needles, deep injections, foreign material in the wound. It all creates those conditions that C. tetani loves.
A reminder that prevention is key, both through vaccination and harm reduction efforts. So we've covered quite a bit of ground already, focusing on 4 key Clostridium species: C. perfringens, C. novii, C. botulinum, and C. tetani. We've talked about their characteristics, how they cause disease, the clinical features, and what makes people vulnerable to infection.
It's a diverse group, but they can all cause some pretty serious problems.
Absolutely. Now, in part 2, we'll shift gears and delve into the world of laboratory diagnoses.
Welcome back to our deep dive into the world of Clostridium species.
It's time to head into the lab and talk about how we actually identify these bacteria.
It can be a real challenge.
Oh, yeah. As medical microbiologists, we know that diagnosing a Clostridium infection is a bit like solving a puzzle.
Definitely. You need to know what you're looking for.
So let's start with the basics. Microscopy. When we suspect a Clostridium infection, what are we looking for under the microscope?
Well, they're gram-positive rods, but their shape can be pretty distinctive, especially when they're forming spores.
Ah, the spores. Those can give us some clues, can't they?
They can. For example, C. tetani has this terminal spore that makes it look like a drumstick.
A drumstick?
Yeah, it's pretty unique. But microscopy alone usually isn't enough for a diagnosis.
Right. We need to culture them too, which can be tricky.
Yeah, because they're anaerobes. You need a special setup to keep the oxygen away.
Anaerobic chambers, jars, bags, and special media that these oxygen-hating bacteria will actually grow on.
Exactly. Reinforced clostridial agar, or RCA, is a good one. It's enriched, so even if there's a tiny bit of oxygen around, they can still grow.
So we've got our anaerobic setup, we've got our RCA plates. What's next?
We inoculate the plates with the specimen. Could be a wound swab, a tissue sample, even food, depending on what type of infection we suspect. And then we incubate them anaerobically and wait.
And as we're waiting, what are we looking for? What kind of growth pattern should raise our suspicions?
Some Clostridia have very distinctive features. C. perfringens, for instance, produces this double zone of hemolysis on blood agar.
That's right. It's because of that alpha toxin, isn't it?
Yep. That double zone is a classic sign. And some other Clostridia, like C. novii and C. sordelli, have interesting reactions on egg yolk agar. Their enzymes create these zones of opalescence or clearing.
So we're seeing some suggestive growth patterns, but how do we know for sure which Clostridium species we're dealing with?
That's where biochemical testing comes in. We can test their ability to ferment different sugars to produce certain enzymes, all sorts of things.
It's like putting together a puzzle using those biochemical clues.
Exactly. And of course, we can't forget about molecular methods. PCR and sequencing have really changed the game.
Oh yeah, molecular methods are so much more sensitive and specific.
Right. With PCR, we can target very specific DNA sequences that are unique to a particular Clostridium species. It's fast and accurate, which can be really important when you need an answer quickly.
And sequencing takes it even further. We can get the entire genetic blueprint of the bacteria.
Yeah. Next generation sequencing is incredible. It gives us so much information. We can track outbreaks, understand how antibiotic resistance is evolving. It's really powerful.
So we've got all these tools to identify the bacteria, but what about when we suspect botulism or tetanus? We need to test for the toxins themselves, right?
Right. And for a long time, the gold standard was the mouse bioassay. But of course, there are ethical concerns with that.
I could imagine. Fortunately, there are other methods now.
Yeah, we've got ELISA cell-based assays. They're more sensitive, more specific, and more humane than the mouse bioassay.
That's good to hear.
And with molecular methods, we can actually detect the genes that code for the toxins. So we can identify those toxigenic Clostridia quickly and accurately.
It's amazing how far we've come in our ability to identify and understand these bacteria.
Oh.
But identifying them is only part of the story. We need to understand how these infections happen in the first place.
Right. The epidemiology is really important.
So where do these bacteria come from? How do they spread? I mean, we've said that Clostridia are everywhere. Soil, water, even our own guts.
They are. Their spores are all over the place. But remember, most of them are harmless. Only a few species actually cause problems for humans.
Okay, so focusing on the pathogenic ones, how do they get into our bodies and cause infection?
It depends on the species. With C. perfringens, it usually enters through a wound that's been contaminated with soil or feces.
And then it finds itself in this anaerobic environment. Lots of damaged tissue. Perfect conditions for it to grow and start releasing toxins.
Exactly. And C. tetani, the one that causes tetanus, also enters through wounds, especially deep puncture wounds.
Where there's not a lot of oxygen.
Right. And anything in the wound, like dirt or debris, just makes things worse. It gives the Clostridia a place to hide and multiply.
C. botulinum's a bit different, though, isn't it? It's not the bacteria itself that's the main problem. It's the toxin.
Right. C. botulinum can cause botulism in a few ways. Foodborne wound and infant botulism.
And with foodborne botulism, it's usually from eating food that contains the preformed toxin.
Right, like those home canned foods we talked about. The bacteria grow in the jar and produce the toxin, and then someone eats it.
And those low-acid foods really the problem, aren't they?
Yeah. C. botulinum loves those low-acid environments. And then with wound botulism, it's often linked to injecting drugs.
Contaminated needles, lack of oxygen at the injection site. It's a recipe for trouble.
It is. And with infant botulism, it's those spores getting into the baby's gut.
Where they can grow and produce toxin because the baby's immune system isn't fully developed.
Exactly. So we've got these different Clostridia with different ways of getting into the body, but they can all cause serious infections.
Which is why prevention is so important.
Absolutely. We need to understand how they spread so we can stop them.
Okay, so let's talk about prevention. What can we do to reduce the risk of these clostridial infections?
Well, the best approach depends on the specific species and how it spreads. For wound infections like gas gangrene and tetanus, good wound care is essential. Clean the wound, thoroughly remove any debris, and if it's a deep wound, surgical debridement might be necessary.
And of course, vaccination is crucial for tetanus.
Absolutely. The tetanus vaccine is very effective. It's really reduced the number of cases. Everyone should be up to date on their tetanus shots.
What about foodborne botulism? How can we prevent that?
It all comes down to proper food handling and preservation. Wash fruits and vegetables, cook food thoroughly, and use safe canning techniques, especially for low-acid foods.
It's all about making sure those bacteria can't grow and produce toxin.
Exactly. And don't forget about honey. Never give it to babies under a year old.
Because of those spores.
Right. Their little guts can't handle them. And for people who inject drugs using sterile equipment and accessing harm reduction services can make a huge difference.
It's about minimizing the risks. So we've covered prevention. What about treatment? What are the mainstays of therapy for clostridial infections?
Antibiotics are usually the first line of defense, especially for wound infections like gas gangrene.
And metronidazole is a common choice, isn't it?
It is. It's a good antibiotic for anaerobes. It can get into the tissues well and reach those anaerobic areas where the bacteria are hiding.
But metronidazole won't work against the toxins themselves, will it? Like with botulism and tetanus.
No. For those, you need antitoxins.
Which is?
Antibodies that specifically target the toxins and neutralize them.
Like a guided missile going in and taking out the threat.
Exactly. For botulism, we have specific antitoxins that can bind to the botulinum toxins. But they work best if you give them early, before the toxins have had too much time to bind to the nerves.
And for tetanus, we have tetanus immune globulin or TIG.
Right. TIG provides passive immunity. It's often given to people who have a tetanus-prone wound and haven't been vaccinated or haven't had a booster in a while.
So it helps to neutralize any toxin that's already circulating.
Yep. So we've got antibiotics to fight the bacteria and antitoxins to fight the toxins, but that's not all.
Right. Supportive care is important too.
Absolutely. Especially with severe infections that can cause a lot of damage.
Like with gas gangs, surgical debridement is often necessary.
Yeah, you need to remove that infected tissue. It helps control the infection and stop the toxins from spreading.
And for botulism, which can cause paralysis of the breathing muscles, mechanical ventilation might be needed.
Right, to help the patient breathe until the toxin wears off.
So it's really a multi-pronged approach, isn't it?
Yeah.
Antibiotics, antitoxin supportive care.
It is. And the specific treatment plan always depends on the patient, the infection, and the resources that are available.
And as medical microbiologists, we play a key role in guiding those decisions.
We do. We help the clinicians choose the right tests, the right treatments.
Well, listeners, we've covered a lot of ground in this second part of our deep dive. We've gone from the nitty-gritty of lab diagnosis to the broader picture of epidemiology and treatment.
And in the final part, we'll put your knowledge to the test with some exam questions. So get ready to see how much you've learned about these fascinating Clostridia.
All right, listeners, welcome back to our deep dive into the world of Clostridium species. It's time to put all that knowledge to the test. Are you ready for some exam questions?
I hope you've been studying because I've got some tough ones for you.
Let's see what you got.
Okay, first question. Describe the mechanism of action of the alpha toxin produced by C. perfringens and its role in the pathogenesis of gas gangrene. Think back to our discussion of how this toxin works on a cellular level.
What makes it so destructive?
All right, let's break it down. The alpha toxin of C. perfringens is a phospholipase C.
And that means?
It's an enzyme that breaks down phospholipids,
Which are?
key components of cell membranes. So basically, this toxin attacks the very structure of our cells.
Ah, okay. Makes sense.
And to be more specific, it targets phosphatidylcholine. That's a major phospholipid in cell membranes. And when the alpha toxin breaks down those phospholipids, it causes cell lysis and tissue destruction. And that's what sets the stage for gas gangrene to develop.
So it's like the alpha toxin is a demolition crew going in and destroying those cellular walls.
Perfect analogy. Okay, ready for question number 2.
Hit me.
What are the main differences between classical foodborne botulism and toxico-infectious botulism? And give some examples of each type. Think about the different ways that botulinum toxin can get into the body.
Okay, I think I've got this.
Time's up. Let's see how you did. With classical foodborne botulism, you're ingesting the preformed toxin. It's already in the food you eat.
Like with those improperly home-canned foods we talked about.
Exactly. The bacteria grow in the food, produce the toxin, and then someone eats it. But with toxico-infectious botulism, the spores get into the body and then germinate and produce toxin.
So the bacteria are actually growing and producing the toxin inside the body.
Right. And there are a couple of ways this can happen. With wound botulism, the spores get into a wound. And with infant botulism, infants ingest spores that then colonize their gut.
Okay, that makes sense. Two very different scenarios, but both involving that potent botulinum toxin.
All right, on to question number 3. This one's a bit more challenging. Explain how tetanus neurotoxin, or TENT, causes spastic paralysis and contrast its mechanism with that of botulinum toxins. So we're talking about two neurotoxins, but they have very different effects on the muscles.
This one requires some deep thinking.
Okay, time's up. Botulinum toxins block the release of acetylcholine at the neuromuscular junction.
Right, which is how they cause that flaccid paralysis. The muscles can't contract.
Exactly. But TENT works differently. It travels all the way to the spinal cord.
To the spinal cord.
Yeah, and once it's there, it blocks the release of inhibitory neurotransmitters, specifically glycine and GABA. And these neurotransmitters are important because they regulate muscle activity. They're like the brakes that prevent the muscles from overcontracting.
So by blocking those signals, 10T is basically cutting the brakes.
Precisely. And without those inhibitory signals, the muscles go into overdrive. You get muscle rigidity, spasms, and that classic lockjaw.
It's amazing how one tiny toxin can have such a dramatic effect.
Okay, 2 more to go. Ready for number 4.
Bring it on.
Why has there been a recent increase in C. novia and C. tetani infections among people who inject drugs? What's going on there? Think back to our discussion about the conditions that these bacteria need to grow.
I think I have an idea.
Let's hear it. Did you get it right? A lot of it has to do with the practice of skin popping.
Skin popping.
It's when drugs are injected into muscle or subcutaneous tissue instead of a vein, and that creates these low oxygen pockets that C. novi and C. tetani just love.
It creates that anaerobic environment that they need.
Exactly. And those spores might be present on contaminated drugs or on unsterile needles, so they get injected directly into that perfect environment.
And remember you said that using citric acid can make things worse.
Oh, yeah. Citric acid is often added to drugs to help them dissolve, but it can also damage tissue, and that just creates even more favorable conditions for these bacteria.
So it's really important to educate people about the risks of skin popping and to provide access to sterile injecting equipment.
Harm reduction is key. All right, last question. This one's about genomics. How has genomics contributed to our understanding of Clostridium species and their pathogenicity? How has this technology changed the game?
Genomics has been a real game changer, hasn't it?
It has. Think about how sequencing those bacterial genomes has given us so much information.
All right, time's up. What did you come up with?
Genomics has allowed us to identify all those virulence factors that these bacteria use to cause disease. We can see the genes that code for the toxins, the enzymes, the adhesins. We can study them in detail.
So it's like having a blueprint of their weapons.
Exactly. And that information helps us develop better diagnostic tools, track outbreaks, understand how they cause disease. It can even help us design new treatments.
It's really incredible how far we've come in our understanding of these bacteria.
It is. And genomics is also helping us understand antibiotic resistance, which is a huge problem. By studying their genes, we can see how they're evolving, how they're becoming resistant to the drugs we use to treat them.
Well, listeners, that brings us to the end of our deep dive into the world of Clostridium species. We've covered a lot of ground, from the basics of their biology to the latest advances in genomics. We've talked about how they cause disease, how we diagnose and treat them, and most importantly, how we can prevent these infections from happening in the first place.
It's been quite a journey.
We hope you've enjoyed this deep dive and learned something new along the way.
Remember, knowledge is power. And the more we know about these fascinating bacteria, the better equipped we'll be to protect ourselves and our patients.
Absolutely. Until next time, keep exploring the microscopic world and stay curious.